Sorry, I just had to restart my recording for a second. So when we fuse the genetic material of a cell with N chromosomes and a cell with N plus 1 chromosomes, the resulting cell has 2N plus 1. This is an abnormal cell. It has 2N plus this extra 1. You can probably guess over here on the right, if we fertilize a cell, we will end up with 2N minus 1. So whatever chromosome is missing here will be missing in the zygote. Now, why that's important? Remember that from that one fertilized cell, we get every other cell in that organism. Okay, it just keeps dividing exponentially. Okay, so any errors in here will be carried over to every. So I had to restart my recording again. So any error in this first cell will apply to every other cell in this organism. Okay, so it's not just that one cell is abnormal. All of those cells, the somatic cells as well, will be abnormal. So, using that example of a fertilized cell that was formed from a gamete with N plus 1, okay, we can have trisomy. Tri meaning 3, so we have 3 copies of one chromosome. And remember, non-disjunction normally only happens to one chromosome at a time. So this is the formation of one gamete, and this is where one of those gametes is receiving both chromosomes. Okay, so it has an extra copy of one of those chromosomes. The karyotype, depending on what your organism, if it's male or female, but it'll have 47 chromosomes. Remember, 2N in a human is 46, so 2N is 46 plus 1, 47. Male or a female, doesn't matter. And then plus whatever the chromosome is. So in this case, if we have 3 copies of 21, it'd be plus 21. Okay, so autosomal examples are Down syndrome, Patau syndrome, and Edwards syndrome. And if this happens with a sex chromosome, if you get 2 X's instead of 1, we have a syndrome called Klinefelter's. Now, Down syndrome is really the only one here where people can live to maturity. Patau syndrome and Edwards syndrome both result in severe birth defects, and in most cases the fetus will die before birth or very shortly after birth. Okay, so this error does result in some very dire consequences. Okay, this is an example of what Patau syndrome and Edwards syndrome would look like. You won't be tested on that, but just understand that it's not like a minor issue. It is a big problem for the resulting organism. So this is a video that I will link to. It's on Vivo or Vimeo, I think, and it's basically showing the difference in view of mental illness, such as the illnesses that result from Down syndrome, in a country such as Cambodia, where the general population doesn't have a great idea of mental health. They're not really educated in these types of things. And so the people who live around, I'm referring to the case in the bottom right, by the way, people who live around this family, they don't know how to deal with them. So the child here with Down syndrome gets bullied, they get beaten, even their parents don't know how to look after them, so they get locked in a cage while the mother and father both have to go to work. So I do recommend you watch that. So we talked about what happens if we have the N plus 1 gamete fertilizing an egg or being fertilized by a sperm. Now we can look at what happens with our N minus 1 gamete. Okay, so also formed from non-disjunction. So this results in monosomy, mono meaning 1, so we have 1 copy of a chromosome. Okay, now there is only really one example of this, and it can only happen with a sex chromosome. Really, you can just have monosomy of the X chromosome. So instead of being XX, you are just X. Okay, 45X. The reason is because it's not that those non-disjunctions don't happen. It's just that we never see them in a fetus or a person because everything else is fatal. So if it happens in your autosomes, if you're missing 1 copy of chromosome 2, 1 copy of chromosome 3, whatever it is, you will not be able to be born. Okay, I don't even know if these progress much further than fertilization. So the one example is 45X, resulting in Turner syndrome. And what Turner syndrome is this case on the right. Okay, you'll see that while it's not a fatal disease, it does cause several abnormalities and also conditions. Okay, so things like this one to do with the aorta. Okay, there are abnormal pigments. Okay, I don't know what this is. Lymphedema, so it'd be peripheral, so swelling of the lymphatic vessels in extremities at birth. Okay, so it's not just sex characteristics that get affected, but it's also general body characteristics. Over on the left, we have Klinefelter syndrome, so it was XXY, that's a trisomy condition, and you will see that extra X chromosome does result in more feminine characteristics, okay, such as breast development, female-type pubic hair pattern, and a diminishing of some male features, such as smaller testes. Okay, there is also some more general abnormalities, such as mildly impaired IQ, I think particularly with language or speech development. How we diagnose this non-disjunction, this is what you covered in that activity. Okay, before you're born, we can look at the chorion, so that's one of the membranes around the developing embryo. Okay, we can sample some cells from that, or we can look at the amniotic fluid. So within this sac, within this membrane, there's a whole bunch of fluid, and that fluid will contain some cells from the fetus. We can also look at the mother. Okay, so now we're not able to karyotype anymore. Okay, with these two examples up here, we can obtain cells from the embryo, and we can do a karyotype analysis. With the mother, though, we can't get cells of the fetus from the mother, but what we can do is we can look at her blood and see her hormone levels, because some conditions cause a change in the hormone levels of the mother. And so just by looking at those hormone levels, we can infer what abnormalities there may be. In an adult, it's pretty simple. You can just give blood or a tissue sample, and then they can do a karyotype from there. Okay, now briefly I'll just talk about some technologies to do with reproduction. So, first of all, about 1 in 10 couples experience problems getting pregnant. Okay, there can be a range of causes. It's roughly a third due to the female, a third due to the male, and about a third are either unknown or both. So we have two main ways of getting around this. Okay, or the two that we're going to look at are in vitro fertilization, what used to be known as test tube babies, and artificial insemination. So in vitro fertilization, what you're doing is you are giving the mother some medicine to stimulate her ovaries to produce multiple eggs. Okay, typically, a woman will only produce 1 egg per month. But remember, if you want to collect those eggs, you will have to go in over multiple months and individually grab those eggs. So you don't want this to be a long process. You want to do it all at once. And so we can retrieve these eggs and take them outside of the body. We can fertilize them with a sperm sample. And once we have some eggs that are fertilized, we can transfer those fertilized embryos back into the mother. Okay, and you were reading it's usually 1 or 2, but sometimes as many as 4 or 5. And then those embryos will hopefully implant and grow into a baby. Artificial insemination is quite less complicated. What we're doing is we're getting a sperm sample from the male. In some cases, if the sperm is of lower quality or of lower count, we can obtain multiple samples, freeze them, and then combine them all into one sample that more resembles normal sperm or semen. So we can concentrate it, and we can use what's essentially a strange syringe to insert it all the way up into the uterus. Typically, sperm is deposited further down into this region, okay, and the sperm then have to swim all the way through the cervix, through the uterus, into the fallopian tube to fertilize an egg. In this case, we're giving the sperm a big head start by depositing them right up in the uterus. Remember that these things don't just apply to humans as well. So there is non-human artificial reproductive technologies. So we can use this to artificially inseminate livestock, pets, zoo animals, or honeybees. So this one bull, Toy Story, has some, I believe it was 50 or 500,000 offspring. Okay, he was a prize dairy bull, and so he has many, many offspring around the world. So by doing so, though, we've decreased the genetic variation of some populations of these dairy cows. I'll look at zoo animals in a sec. And what we can do, I just said that we can decrease genetic diversity, but we can also use them to increase genetic diversity. I'll give you an example of that in just a sec. So with zoos, we have isolated populations of animals all over the world. In some cases, only like 4 or 5 of them. If we keep breeding those animals together, we will end up with inbreeding, and that will cause many defects, and we don't want that. So with pandas, for example, what we can do is we can obtain a sample of panda sperm or panda semen. We can ship that across the world and essentially breed this panda with another panda somewhere over the world, like on the other side of the world, so we can increase genetic diversity. Why I put that up is because some animals need artificial reproductive technologies to reproduce. These pandas, for example, aren't actually able to mate with each other naturally. Okay, pandas have a notoriously low sex drive, are incredibly difficult to reproduce in captivity, but because of artificial insemination, these two have produced 2 cubs and may be producing a 3rd. There was actually even a case where they did this with a deceased panda and managed to, I think, this deceased panda managed to father some offspring. So by doing so, okay, by shipping gametes around the world, we can increase the genetic diversity in a population. This is just an abstract from an article which was looking at the founder of increasing genetic diversity in founder populations. So that's very small populations that are in a new environment usually, so there may be only a handful of them. But because of that, we have very low genetic diversity. So in this case, they can use applied reproductive technologies to reproduce this small population with a larger population of those animals, or even from with genetic resource banking, we can even use gametes from organisms that have passed, so from previous generations, to maintain the genetic diversity of that population. Okay, so with ART, we can increase genetic diversity in those cases, but we can also decrease it with examples of such as this dairy cow. So hopefully now you can meet these objectives. So define non-disjunction and how it occurs, how we diagnose non-disjunction, and the use of artificial reproductive technologies.